This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 215, recorded on January 11th, 2013. Hello, everyone. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How are you doing there?
Doing okay. It's clouding over, and we're supposed to get sloppy weather this weekend, but other than that, things are all right.
Very cloudy here. Very, very much.
Yeah, 34 Fahrenheit, 1 Celsius.
You've succumbed. You give both.
Yeah, now I give both.
What do we got here?
I don't want testy emails from people.
Testy. 6 degrees Celsius clouds. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
How are you?
Hey, Kathy.
Fine. We are having a heat wave. It's 51 degrees Fahrenheit, 11 Celsius.
Wow.
And it's going to be even warmer tomorrow. Yeah.
It's climate change, right?
Yeah.
Well, it's not a heat wave like they're having in Australia, apparently.
No.
It's summer down there, right?
It's summer down there, and their weather service had to change the color coding on their maps because they're hitting temperatures that they hadn't allowed for before.
Wow.
Okay, here's a question for you. In 2012, Sandy was a disastrous weather condition. But what was an even more disastrous weather condition in 2012?
The drought?
Exactly.
Yeah.
Yeah, on New Year's Eve we had a quiz on events of 2012, and that was one of the questions. Yeah,
it's the stuff that does the most damage is usually the events that go for a long period of time over a wide area, not these acute big storms.
Yeah. I don't know where did that come from. Anyway,
weather.
It's getting warmer.
Yes, it's getting warmer.
All right, we have a few follow-ups from last time. The first is from Patty, the author, the PI on the Racaniello raccoon virus paper. Raccoon polyomavirus. Patty, so sorry I missed you for this show. I've just listened. I would have had great fun talking with you all about this RAC-Pi virus, but I missed my chance. Thankfully, you all did a great job. Yeah, I guess. If we had done a bad job, she would have felt badly. Then, back at work, and just as a follow-up for the conversation about raccoons, 1, completely agreed about the proof needed for causality. 2, Dr. Imperiale helped us in this story by providing constructive feedback on our paper. He was very interested, and it helped me to understand where the viral oncogenesis field stood on this. He was gracious about his doubt, just as he was in his comments about large T expression being scanty. Heavens, it is that. But IHC immunohistochemistry is a pretty blunt tool, and we're using a Merkel cell polyclonal antibody that only vaguely maps to the raccoon polyomavirus genome. Those are some possible reasons for the lack of robust signal. In the meantime, we've cloned large T into an expression vector so we can make more robust antibodies in the future. Let's see. The CT counts from our real-time PCR assay. All right, so the real-time PCR assay tell a different story. So I guess she's getting higher large T by real-time PCR.
Right.
3, the dogma. We, of course, thought that we could help ourselves to that end by doing Southerns to prove integration, which has become, since Merkel cell virus, somewhat of the dogma for causality. But as you noted, raccoon polyomavirus, at least the bulk of it, is unintegrated. There are more sensitive ways to assess integration at low levels, but I think we have to consider persistence in the absence of integration. That's where we are. For me, it's just plain fun to consider it, and I could use a real virologist's help as long as they are not too dogmatic in this. As a pathologist who is either 1, thinking outside of the box, because she is outside, or 2, unencumbered by facts, which I am, it seems like there are some possibilities here. Take mitochondria. They have a circular, like polyomavirus genome, and regulate their replication, transcription, and translation completely independently of the nucleus. I look at ultrastructure all the time, and many viruses are associated with mitochondria. Also, couldn't we be dealing with low-level replication, encumbered by something other than an integration late gene deficiency deal? You're the best at questioning dogma. What do you think? And 4, the species jumping end host possibility. We have found this virus in an opossum and a squirrel. We don't know if it's replicating there. Given that other polyoma viruses can become replication deficient and oncogenic exactly by jumping hosts, we thought this should be a consideration. We're on it and looking at humans and other mesopredators in the mix. What a wonderful show title.
Mesopredators in the mix. Yes.
That's great. It's like Gorillas in the Mist.
Gorillas in the Mist, yeah.
Is it possible that Patty actually... No, she can't be the Alan Dove of pathologists. And she finishes, and from your friendly neighborhood veterinary pathologist, so much roadkill, so little time. You know, she's got 2 great episode titles.
2 great episode titles there. It's really unfortunate we didn't have her on the show.
Would have been great, and she has some very nice ways of putting things here.
Yeah, and some
box.
Some good additional observations here too. This is very interesting about the possum and the squirrel.
Yes, very much. I think when Patty has more data, we'll have you back.
Yeah.
Or not back because you weren't here, but you were here in our thoughts.
Yeah.
We'll have you on. I bet it would be fun. And you shouldn't be encumbered by facts. Remember, Stuart Firestein says ignorance is what drives science, and he's right. And so you are doing just fine there.
It's useful to know certain facts, but not to be encumbered by them.
Yes. I don't see why it would have to integrate. I mean, there are other ways. I mean, the goal is to suppress lytic replication so the virus doesn't kill the cell, and then constitutively express large T so you drive replication of the cell, and that's how it gets oncogenic. And there are other ways to do that. It doesn't have to be the same as all other polyomas, right?
Yeah.
So I think it's good to think outside the box. Don't say, this is a polyoma, it has to work this way. I think you're doing the right thing, Patty. What is a mesopredator?
A mesopredator is a medium-sized predator. So Wiktionary says that includes raccoons, skunks, snakes, cats, and foxes.
So would that make Alan a mesopredator? Because he's not really tall?
Well, is he a predator?
Oh, sure. You should have seen him in the lab. Wow. I never heard that name. It's a good one.
It's a useful term.
Mm-hmm.
But
sort of everything in that mid-size. I think humans would probably be, what would macro or mega? No, macro predators, right? Or
macro predators.
And would small, really small animals, I don't know, like praying mantises, would they be micro predators?
Yeah. Is there such a term?
I don't know.
In Wiktionary? How do you say that?
Wiktionary?
Wiktionary.
Micropredator.
A micropredator is a faculty in a microbiology department. Wow. I really liked it. Could we use the title even though it has nothing to do with the episode? Okay. We have another one from someone called Anonymous. Dear all, I am a long-time fan of the show and I'm very grateful for what you do. Quick question regarding your discussion of the Factor X adenovirus story. If Factor X has evolved to combat some adenovirus species, does this mean...
That's Factor 10, by the way.
Yeah, I'm sorry.
It would be so much cooler for it to be Factor X. I agree totally. But, yeah.
The thing is, I really get on people when they say OS X. And so here I'm doing the same thing. Factor 10. It would... does this mean that adenovirus or some other virus with similar capsid properties has exerted pressure on the germline such that those organisms that produce factor 10 that binds adenovirus capsid have a selective advantage? Alternatively, could it not also be possible that some adenoviruses derive a benefit by binding to factor 10? Perhaps binding to factor 10 allows entry into a beneficial cell type. I think they showed that in the paper, right?
Yeah.
Factor 10 plus that allows liver cell entry, right?
Correct.
So that allows, and if you mutate the 10 binding site then doesn't get into the liver, right?
Right.
All right. So that's good for gene therapy, if I remember that, them saying in the paper. So it doesn't kill you by getting this massive inflammatory liver reaction. Or perhaps binding to factor 10 slows down the kinetics of infection, thereby allowing for long-term advantages by promoting persistence. These alternative possibilities seem equally likely to me, since it seems the virus would have enough evolution power to evolve away from factor 10 binding before the mammalian genome evolution could catch up. In general, it seems that authors have to sell the host defense side of these newly identified interactions that slow down replication, when it is just as likely, more likely, that persistent viruses derive fitness benefits by allowing for an attenuated infection. Again, keep up the good work, and many thanks for being so selfless with your time.
So the first thing he raises the possibility of whether factor 10 has evolved to combat some adenovirus species. I suspect it's hard to really know what the factor evolved for, but my suspicion is it's more likely that it developed as a coagulation factor, and then some of the adenoviruses maybe took advantage of that to expand their host range and be able to get into liver cells or something. But we can never really know about the evolution of that, I think.
So you think then when did the innate stimulation come in? At the same time, I guess, right? That add bound to it so that it could get into a new cell?
Well, we don't know that factor 10 binding correlates in every adenovirus with this innate phenotype yet. So if there were one-to-one correspondence with that, then maybe you could say yes, that they evolved together.
And the idea that it would slow down replication, and that's a positively selected trait, that's not a bad idea, right? Because persistence is, as we will see in one of our papers today, persistence is a feature of many viral infections. So, yeah, I think you're thinking outside the box, anonymous.
Definitely.
So I thought of the idea of positive selection here. So is there an arms race between adeno and the host, right? So as he says, has it exerted pressure on the germline? So you can imagine that adeno, it may be mutating to avoid binding factor 10, and then the host changes. And you can see this if you look at sequences of factor 10 through phylogenetic history. So I emailed Harmit Malik about that, who's the king of positive selection, genetic conflict. And he said, we have not looked, but you might expect, since it's involved in host recognition. And then his postdoc, Matt Doherty, who was on TWiV some time ago, said he looked and he didn't find a strong evolutionary signature. In other words, no strong evidence for positively selected amino acids, and nor did he think there's any evidence that AD has been antagonizing this interaction. So probably not. But that can happen, right? Anytime you have a host virus protein-protein interaction, there could be a genetic conflict there.
Right.
There are lots of examples of that. Okay.
Yeah, although in the case of adenovirus, it's a pathogen that's evolved to be pretty mild most of the time. It's not going to place huge pressure on the host, and the host may not place huge pressure on the virus. They may have reached an equilibrium at this point.
All right, we have two papers. Well, we have one paper and one smorgasbord today.
Yes.
If anyone's hungry. First paper is in PLOS pathogens. We had a guest here yesterday by the name of PLOS, P-L-O-S-S.
Really?
Yes. I didn't ask him if he liked to publish in PLOS.
Wow.
But this paper is quite interesting. It has to do with rabies virus, which we often get a lot of questions about. And we happen to have one actually later today. The title is Immune Clearance of Attenuated Rabies Virus Results in Neuronal Survival with Altered Gene Expression. And Kathy, you picked this one up. I guess Do you look at rabies papers, or you just caught your eye?
No, this one just caught my eye somehow. Yeah. And we had talked about rabies in neurons recently somehow, so I think that's why. Yeah.
One of the authors is Glenn Rawl, in addition to Gum, Werblick, Adya, and Schnell. And Glenn was on the paper, or the PI of the paper that you and I did a long time ago on dually infected mice. Remember that? Right.
And then you have Matthias Schnell.
Yeah.
Did you have him on the show, or somehow you have him on the webpage?
We had a question, and Rich had emailed Matthias, and we got an answer. So he's in the show notes there about rabies. So we have talked about rabies a lot, as everyone knows. This is a virus that causes a very, very highly fatal disease. If you get rabies, man, you're probably going to die. Most people do, and very few have survived. Especially
when you develop symptoms.
Yeah, when you develop symptoms. If you have not been vaccinated, very, very few people survive. And if you remember, we talked about the Wisconsin protocol as one way of having a few people at best survive. It's a rather radical protocol.
But there's a long window between, often a long window between exposure and onset of symptoms during which the vaccine is effective.
Yeah, so you can have, I think they cite in this article, between weeks and even a year. Depending on many factors, where you've been bitten, how much virus you get. The central question here is what causes the symptoms of rabies, which everyone knows: aggression, hyperactivity, muscle weakness, paralysis, coma, death. And I think we touched on this before. There's some evidence that it's not really killing of neurons that's doing this. And in fact, they say in this paper that when they have looked at brains after people have died of rabies, there's very little evidence for tissue damage and neuropathology. So the idea has evolved that there's some other mechanism, some non-cytolytic mechanism involved. And that's what this is, some alteration of neuronal function. Okay, and that's what's causing all the symptoms of rabies. So that's they make a very interesting experimental system in this paper to answer this question. Really, really cool. They make a mouse which has been given a transgene that allows for the production of green fluorescent protein constitutively.
Right? Oh, no, it's
red fluorescent constitutively.
Red, TD tomato.
Okay, so they make tomatoes.
Red fluorescent protein. They make tomatoes. No, they make a fluorescent red protein.
So they make red protein constitutively, and then upstream of the red protein gene is a GFP gene, which is only produced if the tomato is upstream of the red is upstream of the green gene in this construct, and the red is flanked by what are called LOX-P sites. These are sites for a nuclease called CRE. And so you only get green expressed from this, but when the red is you only get red expressed from there, but when the red is excised by CRE nuclease, then you get green expression. So I've confused everyone. I'm very sorry about that.
Right. So this LOX-CRE system has been used pretty widely in recent years, and it's borrowed from a bacteriophage, right?
That's right. It is.
And part of this phage's normal life cycle is using these LOX sites, and then it has this recombinase called CRE that specifically recognizes the LOX sites and recombines them, which loops out any piece of DNA that's in between them.
It's used widely in all sorts of studies to turn
this comes
genes
up all the time
on and off. Yeah,
it's so incredibly handy to be able to do this. So you put these LOX sites on either side of some gene that you want conditionally deleted.
Right.
And then when the CRE protein gets introduced by whatever mechanism you're studying, it loops out that piece of DNA. And in this case, they've got this really clever adaptation of it where when it loops out the red, the TD tomato gene, what you're left with is a promoter that drives green fluorescent protein.
Right.
And that's a permanent change in the DNA of the cell. So when this happens in a cell, the cell switches from glowing red to glowing green, and it stays that way.
So you're saying that tomato lox and bagels make a good combination.
Yes.
Okay. This is a good reason why we should be doing basic research on things like bacteriophages, because you never know when something like this is going to pop out of it.
Well, and green fluorescent protein comes from a jellyfish.
Right.
And do you think red tomatoes come from tomatoes? Probably not.
Yeah. But just to set the record straight, I believe they did not make these mice themselves. It looks like they purchased them from Jackson Labs.
Right.
Yes, these are commercially available mice that you can get that have now there are bunches of strains of these. You can get different things flanked with lox sites, and this is one of them.
Now the Cree that is used to excise the red tomato gene is provided by rabies virus that they've constructed. So you can imagine now you're going to infect with rabies virus, and then whatever cell that it infects is going to be permanently converted from red to green.
Right.
As long as it doesn't kill the cell, right? And so they use an attenuated rabies virus that will not kill the mouse or the cell, so that they can do these studies. Of course, if they used a virulent rabies, it would wipe all the mice out. You couldn't do anything. So the point is to say, are there going to be cells that persist in the mice for a long period of time that have been infected by rabies, and we're going to use the switch from red to green to detect that?
Yeah, because the argument is there's this controversy about whether you're getting neuronal death causing problems in rabies or neuronal malfunction.
Right.
And the problem is that when you take apart a mouse and you look in its brain, you can't tell which cells were infected and which weren't.
Right.
So they've built an indicator where once these cells get infected with this Cree-expressing virus, the cells will then turn from red to green and stay that way. So you can identify any cell that has ever been infected with this virus within the brain of a mouse.
Okay. So they make this virus, they characterize it. In cell culture, they show that it doesn't have a replication defect. It can convert in cell culture red cells to green, so it seems to work. And then they put it into mice. They actually put it in intranasally. And the mice get very typical symptoms of rabies without paralysis. They get weight loss, fur ruffling, ataxia, hunched posture, which are all things I do on a daily basis. But none display hind limb paralysis. And many of them, 60 to 70% of the mice survive so that they can study them long term. So they look on a long-term experiment up to 6 months post-infection, and they sacrifice mice at different times. And they can look in the brains and see, are there green cells? Because that's the mark of rabies. You can make a title out of something like that, Alan, right? And they also show that in these green areas, they can stain for one of the rabies proteins, which tells you that virus has been there. So you're not just spontaneously going from red to green. You have to have virus present to deliver the Cree nuclease. So they see that there are green neurons in mice for up to 6 months.
And it looks, when you're looking at the brain sections on this at lower magnifications, it's almost like a plaque assay.
It's amazing.
You've got these spots of cells that are green spots inside the red brain. By the way, this paper is open access, so everybody can go and look at the figures themselves.
That's right. Even Dr. Ploss.
Even Dr. Ploss could go and look at it.
So they see throughout the brain, cerebral cortex, hippocampus, cerebellum, all green cells. So in other words, these are presumably where rabies virus has been throughout the brain.
Right.
And that's, in fact, what we know about rabies infection, that it will go throughout the brain.
But these infected cells are still alive.
Right.
How would you know that? Because they're making green fluorescent protein?
Right. They're still synthesizing the protein. And the higher magnifications, they look like neurons.
Yeah.
There isn't gross pathology going on here. You've just got these green spots and now surrounded by red tissue, and the only difference you can tell is that some of the cells are green and some are red.
Right. That's a good point. So there's no overt pathology in the brains.
Right.
You can't really tell that rabies has been there.
Even though the mice demonstrated the symptoms of it.
Right. So they had initially these symptoms, and I guess they went away and they began gaining weight again, right? So they recovered. And there is a clearance of virus over time, although the RNA does persist for quite a while. They have 1,000 copies of the genome as late as 6 months after infection.
Mm-hmm.
And that's surprising to them, and they speculate that. There's some viral nucleocapsid protein that's stabilizing the RNA in there. There's no evidence that this RNA is replicating. They don't find any of the one mRNA and mRNA that is needed for replication. So the genome is just sitting there. So the virus has infected cells. It's caused symptoms, and then apparently replication has been dampened, and then the mice have recovered, but the cells are still marked by green. You can see that they're there for a long period of time. So the question they want to ask is, are there changes in these neurons? You can't see any changes, but can we look at the genes that are expressed in these neurons and see if there's some change in the pattern of mRNA expression? So they purify neurons from these mice by flow cytometry, and then they do a microarray analysis where they hybridize to slides containing representative oligonucleotides from many genes, in this case thousands of genes. And they can say what genes go up and what go down in these neurons that have once seen rabies virus.
And one of the cool things about this system is because they've tagged the previously infected and the uninfected cells with different fluorescent colors, they can sort out cells from the same mouse.
Mm-hmm.
So you can take one mouse and grind up the brain and sort the cells, and you get the infected and uninfected from the same mouse. So you're not dealing with quite as much noise as you would normally see in a gene expression assay,
right,
because it's the same organism. It had exactly the same conditions for these cells.
Right. That's right. So that's within the mouse, the control of uninfected versus infected, and they actually do microarray with both of those, right?
Right.
So they find 1,248 genes that are differentially expressed. The fold change is not high, ranging from 3-fold upregulation to 2.7-fold downregulation. Only 127 genes differed by more than 2-fold. But you don't need to have a lot of genes changed, I would think, to alter a neuron.
Right.
So you don't need hundreds and hundreds. So 127 is pretty good. And they can classify these in different areas of different functional areas, like cell-cell signaling. Cell-cell signaling was the most significant function impaired, and then genes involved in nervous system and behavioral development were another one.
Behavior. This very general behavior category was at the very top, and then cell-cell signaling.
Yeah. So you can imagine that changes in those. They can't prove that any of these changes are causing rabies, of course,
right,
not in this paper, but this gives you something to look at in the future. You can imagine, playing with these genes and seeing if you get some pathologies and so forth.
I thought it was interesting that the name of the bioinformatics analysis was called Ingenuity Pathway Analysis.
Yeah.
IPA.
IPA. Beer.
Yeah.
So I had a thought here, two thoughts or sort of questions. One is, since you can only, when you do these sections at different times after rabies infection, you're always looking at a different mouse, obviously, because you're taking the brain out and section it. I would like them to do some kind of non-invasive imaging, so they could take a single mouse and look at it at different time points. Because how do you know that some neurons aren't dropping out? So we don't have any quantification of recovery in terms of neurons. These cells are marked, but maybe there are other cells that died, right?
Right. You're only seeing the cells that survived.
Yeah. Now, I can understand that probably a lot of cells did not die. Otherwise, you would see pathology, right?
Right.
But maybe some did. Can you get very neuron specific in imaging? Could you go in and always look at the same cluster of neurons in a brain by that?
It can be done in some systems. There's I did a story on I've done several stories on live imaging, and there's this sort of convergence going on with imaging technologies like MRI and PET scans, and also confocal microscopy and two-photon microscopy, and even to the point of people installing a little window on the, making a hole in the mouse's head and installing a little window on it to be able to look through. The problem with a lot of this, especially with brain imaging, which is obviously of huge interest right now, is the optical techniques are usually not very deep.
I see.
You can't see down. I mean, the brain is so opaque, and the other types of techniques are not very high resolution or haven't been very high resolution, but it's getting better. So we'd have to talk to somebody who's working on that. But my feeling is that there are probably ways to get close to this, and maybe assay. You might have to use some different marker rather than a fluorescent marker. Maybe something that would show up in a molecular imaging study, a 3D molecular imaging study. But this paper certainly outlines that it's possible and shows that you've got a lot of surviving cells that got infected and cleared the virus or are clearing the virus. So yeah, I would think that would be a direction to push this in, definitely.
I don't know what kind of numbers we're talking about, I mean, they say there's no pathology, but how many dead neurons could be below that radar, right?
Right.
I don't know. And could that account for some of the symptoms?
How many neurons does it take to change a brain? Yeah.
I know it takes 5,000 to paralyze a muscle. You have to kill 5,000 neurons in an area that innervate a single muscle to paralyze it. But, how many do you have to kill to get rabies symptoms? I don't know.
Right.
The other question is this mutant virus that they work with. And they admit this, that maybe it's just odd and has nothing to do or little to do with what happens in wild-type rabies. I know they had these...
They did use an attenuated virus, yes.
Yeah. I know they had rabies-like symptoms, right? So that's good for them, but I'm just wondering if that made any contribution. They say they do acknowledge that.
Yes.
Possibility, though.
So if they used a more virulent virus, you would think that they would just have to look much sooner.
Yeah, right.
Well, and what I was thinking is, for the question you were asking, Vincent, if you could look in the brain, I was just thinking that it would be hard to keep a mouse alive a long time with a window in his brain. But if you used a more virulent virus, then you'd have to be looking at short times after infection anyway, and you might be able to have a mouse with the window in the brain stay alive and do the study.
Yeah, if you were looking at a period of a few hours, you might have more flexibility in what you could pull off for the imaging.
So they say what's the reason for doing this besides understanding how rabies causes disease? They say maybe if we understand what's happening to the neurons, we could devise therapies to fix them, right? So if you know exactly what's going on, maybe you can fix it one day. So instead of having to put people in the cold with whatever that drug was that they do in the Wisconsin protocol, and you do something else.
In a coma. Yeah.
So this is pretty cool. I like this. It's a nice start.
Well, and they also point out that it's useful because a lot of times there are neurological diseases where a viral etiology is suspected, but there's no sign of the virus, of a virus. Yeah. And this goes along with that study that you and I did earlier, Vincent, of Glenn Rawls, is that, maybe the virus doesn't have to be there anymore for you to still have these effects.
You know what disease came immediately to mind?
I don't know.
Alan will know this.
No?
It's one we talked about a lot last year.
Oh, CFS?
Yes.
Oh, yes.
Yeah.
I mean, there's the idea the virus is, people are infected and then the virus is gone, but the mark of the virus is there.
Yeah.
The malady lingers on, right?
The malady lingers on. So this would be a good way to look at that in a model system anyway. Can't do it in people, of course.
Yeah.
Right.
Yeah, and I thought this was actually quite encouraging news, even just for rabies, because you've got a lot of cells surviving infection, which seems like good news. And maybe there's something that we can do to address the changes that have occurred in those cells to try and get them back to normal.
Right.
Because whatever's happening, it may not be that it's just wiping out cells.
Right. No, maybe you can fix the cells in some way.
Maybe you can fix it.
Yeah, you got to figure out what's wrong, though. That's hard because you got a lot of genes here. Then you have to figure out what happened to alter their levels and what they're doing. So there's quite a bit of work.
Yeah, and getting a drug into the brain is a tricky problem, and getting a drug into the brain that does something specific is an even trickier problem.
Yeah, I know. I would argue that the impact on rabies is not going to be huge because there are not a lot of, at least in countries that vaccinate, there are not a lot of fatal cases, but this is going to tell us about brain function.
Yeah.
I think that's the positive. You use rabies as a probe, as usual. Viruses showed away, right?
Yeah.
Okay. Is that a good way to close on this one?
I think so.
Anything else? All right. The other thing I thought we could do is to give everyone an update on this novel coronavirus.
Yeah.
And we talked about a while ago with
Matt Freeman.
Matt Freeman. Man, I'm 60. Oh my gosh.
I heard something about that.
How many neurons does it take to be able to forget a name?
Probably just a few. Oh boy. Anyway, we talked about this novel coronavirus, which was originally isolated from a patient with severe respiratory illness in Saudi Arabia. That was back in September. There was a second case in a Qatari citizen who was brought to the UK. And then from that, virus was isolated, and it turned out to be a new coronavirus, which we'll talk about in a moment. And since then, there have been an additional cases. There's a retrospective look at a series of infections in Jordan in April of 2012. And apparently someone on ProMedMail said, this looks like an interesting outbreak and, severe respiratory infection. Maybe it was this same corona-like virus. And it turned out to be that 2 of the cases were positive for this novel coronavirus. So the original outbreak, there were 11 people and 7 nurses, and so 2 of them were positive.
Right. So they had this outbreak of severe acute respiratory illness, SARI, and they...
it was reported, right?
It was reported, and they investigated it at the time, back in April,
right.
And they tested for SARS, and they tested for a bunch of other things, and everything came back negative. And they just kind of shelved it and said, well, a couple of people died, and we had these infections, and we don't know what caused it. And then subsequent to that, with these coronavirus cases elsewhere, the WHO went back to those cases and said, hey, is it this? And the answer turns out to be, well, at least 2 of those cases that they had samples from were.
Right. So that's
these are PCR findings.
So that's the way this happens. I think most of these respiratory infections, or any infection where you can't figure out the etiology, you never do. And maybe it's a novel virus if you looked. And so here they had the sequence in September, so they could go back and look. Who knows how many times this happens?
Well, then this is a little bit like, say, the Legionnaire's disease Pontiac fever story.
Right.
Where you had a respiratory disease, unknown etiology, and nobody was able to figure it out. Right. And then they put the samples on the shelf, and years later you come back and you find out that was Legionnaire's disease.
Right. Then there subsequently were more cases, all, I believe, in the Kingdom of Saudi Arabia. Late November. So there's a very nice table that summarizes 9 cases total, 5 fatalities, all with severe symptoms. And, there are 2 clusters of infection, but there's not a lot of connection between them as far as we can tell. So, we don't know how they acquired the infections, and still we don't understand if there was transmission among the individuals, as far as I can tell.
And a lot of the people who got this, including some of the fatalities at least, were people who didn't have any known contacts with animal reservoirs that they'd suspected. So it wasn't all pig farmers or people who worked around bats or anything like that.
So I think that's the number so far. As I checked yesterday at ProMed Mail, and I don't see any new cases. So November was the last. So clearly, this is this is not spreading like wildfire, right? This has infected a number of people, but doesn't look like it's yet able to spread like SARS did initially. Right. But WHO is very aware of this, and they got alerts out, and people are looking out for more cases, obviously. Yeah. Not too long after the first few cases, Ron Fouchier's group, everyone must know that name from the H5N1 story last year, sequenced the entire genome of this virus, and it's been called human coronavirus EMC, and EMC stands for Erasmus Medical Center, because that's where they isolated it. But it's unfortunate because we already have an EMCV. And so it's going to be a little confusing, but that's the way nomenclature is. They got the whole genome, and what they find is that, so this is a paper in MBio, genomic characterization of a newly discovered coronavirus associated with acute respiratory distress syndrome in humans. Most closely related to bat coronaviruses, 2 different species of bats, which is really interesting because, it's thought now that the bats are the reservoirs of coronaviruses. So there's a bunch in there and occasionally spill over into other species, including us. But how these individuals caught it is not clear, right? Because they don't have any apparent contact with bats, even though some of them were on a farm where I guess you would find some bats sometimes.
Right. Well, and for, at least for rabies, I think there's evidence of just aerosol spread. Yeah. Maybe it's on fomites or something, and maybe that could be what's happening here, and people are just not aware that they were in contact with it. Right.
So I think the main thing that you get from this paper, one, is that this is most closely related to bat coronaviruses, and next that they can identify all the open reading frames in the sequence, and some of them have obvious homologies with other coronaviruses like the RNA polymerase and the exonuclease and so forth. But there are some that are new and novel. So this is going to be interesting for our virologists to study. And many are jumping on this, as you might imagine, a brand new virus. It's something you can establish your territory with or your reputation and do some interesting experiments. So I'm sure we're going to hear a lot about that in the next years.
Yeah.
They noted sorry, go ahead.
I just wanted to be sure to point out, and I guess we'll link to this Nature news story about Declan Butler, which I think does an outstanding job of summarizing the situation and also addressing most of the major issues that the WHO and CDC et al. Need to look at this point.
Right. Yeah, we will put that in the show notes. I don't know if it's going to be open or not, but.
And one of the things they point out is that at the moment it looks like there's a pretty high death rate, but if there are a lot of people in the vicinity that have antibodies to the virus but haven't had serious disease, then that could alter that, and that's reminiscent of the H5N1 story as well.
Yeah, I think that's actually a really critical point. There's no antibody testing being done at this point, so the positive samples are all PCR positive, which means you find the virus in the sample. And that's why in this retrospective analysis, they found a lot of people had this severe illness, but they were only able to confirm the virus in 2 of the samples.
Right.
Probably, the people who had it and got over it, they got over it, and the virus is no longer in their bloodstream. So I think it's critical to develop an antibody test to find out who's been exposed to this thing.
Yep.
And that might also help explain the pattern of cases, because it seems likely that there's a lot of spread going on, and we're just seeing the cases that happen to be severe.
So I'm sure developing an antibody test is in process. It's not a difficult thing to do. You just have to decide what viral protein you want to use. I guess you could use the glycoprotein. That makes the most sense. You'd have to make some antibodies against it to use as a control, but you basically would make the protein by recombinant methods, attach it to plastic, and then use it to capture antibodies from sera in an ELISA-type format.
Right.
So the first test that was done was the PCR test, and that was used almost immediately based on the sequence. But now, as you say, we need to know what antibodies are out there. The last thing they say in this paper is it is important to develop an animal model that can be used to fulfill Koch's postulates by demonstrating that the isolated virus can cause the observed disease, of course, in animals. Right.
Mm-hmm.
Okay, and the other paper that I thought was interesting is also in MBio. They are coming out like crazy. This one is human coronavirus EMC. Again, that's the name of this new one. Does not require the SARS coronavirus receptor and maintains broad replicative capability in mammalian cell lines. So the receptor for SARS coronavirus is a protein called angiotensin converting enzyme 2, or ACE2. And this is an interesting protein that happens to be present quite deep in the lung, and that has sort of two effects on pathogenesis. First, when the virus gets down there and replicates, you get severe lung disease. But it's harder to transmit because as the virus is released from cells deep in the lung, it's not as easy to cough it out and to transmit it to other people. So in this paper, they ask, does this EMC virus use ACE2? Does it require ACE2 to infect cells? And the way they basically do this is to try and infect cells that either do or do not express ACE2. For example, they use a line of BHK baby hamster kidney cells which don't have ACE2, and they express it using a plasmid transformation, and so now they have a pair of cell lines, BHK with and without ACE2. If you don't have ACE2, these cells will not be infected by the SARS coronavirus. If you do put ACE2 in them, then they can be infected. And the EMC corona will not infect these cells either way. So it's not apparently enough to get this virus to replicate. Now, an interesting point here, I have to make a little complaint. They make a statement. BHK cells normally do not express ACE2 and are not permissive for SARS coronavirus. Now, in the classical use, permissivity has to do with post-entry events in the replication cycle. So if BHKs are not permissive for SARS coronavirus, if you then put a receptor in them, the virus would still not replicate because the cells are not permissive.
Right.
They would become susceptible, but not permissive. So that's not correct use of the nomenclature. Now, I'm being a little picky here because a lot of people don't use it correctly, but many years ago when we were first writing our textbook, I learned that susceptibility has to do with receptor presence and permissivity with internal things. So
so they conferred susceptibility
exactly
on cells to SARS. They did not confer permissiveness because the cells were already permissive for SARS.
Right. So Alan, how would you tell if a cell is permissive for a virus? What would you do?
To tell if it's permissive for a virus, I would, if I had a way to transfect it in, I'd probably try that.
Yes, exactly. That's what I was thinking.
Yeah.
Now, so here's what they say. They infect BHK cells with or without ACE2 with this EMC virus. They conclude that human ACE2 is sufficient to permit infection by SARS, but not EMCV in BHK cells.
Right, and in cells that are susceptible to the novel coronavirus, to EMC, blocking the ACE2 receptor does not block infection.
Right. So later on, they do show that. But right here, from just this one experiment, they really cannot conclude that the ACE2 is not sufficient to permit infection by EMC, because they don't know if the BHK cells are permissive for EMC. Right? Guys?
Okay.
Yeah, so if that were the only experiment in the paper, then you couldn't conclude anything from it because they really need to put the genome into these BHK cells. So from the BHK experiment alone, they cannot say anything about the receptor because they don't know if these cells are permissive for this coronavirus.
Right.
Now, I hate to be picky, but here on TWiV, we get tough.
Well, and details matter.
Details do matter in a case like this. But as you said, Alan, they have other experiments where they show that blocking ACE2 with antibodies that would block SARS do not block EMC infection. They have a few other experiments as well. So antibody blocking. They have some, let's see, cell lines. Where was it? There was one other experiment that deals with the receptor, but now I've forgotten it. Okay, so then they make cell lines from bats, actually, 2 different species of bats. And these cells can be infected by EMC, but not by SARS. In fact, it turns out that most coronaviruses won't infect bat cells. That is, the human coronaviruses. And so it's quite unusual that this EMC is able to infect bat cells, and they also find pig cells and human cells. So it has a very broad tropism, which is unusual. So they think, maybe this virus could infect many species and thereby get a hold and eventually get its way into humans as a consequence of this tropism. So the bat coronas, as they typically come out of bats, they don't replicate in human cells. They need changes in order to do that. But this virus, which doesn't seem to be well adapted to humans, already has a broad tropism.
Right. But that said, these are cell experiments.
These are, yeah.
So we don't know that it actually infects bats or pigs or that it can successfully infect any of these species. But it's suggestive that there may be fewer barriers for this virus than there would be for some other viruses.
Right. Mm-hmm. This breadth of tropism is absolutely unique among coronaviruses. Only once have researchers succeeded in culturing any corona in bat-derived cells. And that's the one experiment that Matt told us about, where they used an engineered virus with a glycoprotein swap. So this is quite unusual. What it means is anyone's guess.
Right.
Right. Mm-hmm. But it's kind of neat. And, of course, they don't know what the receptor is. It's not. This is interesting. It's like a negative result, which in the past I have been unable to publish for polio, but I guess because this is a hot new virus, you can publish negative results. So they don't know what the receptor is. It's not ACE2, but something else.
Well, and there's value in that for other people to not be doing more experiments with ACE2 necessarily. I mean, you want to get that information out there. So there was a long time when people were making transgenic mice and not seeing phenotypes and not being able to publish things. And eventually there became a way to do that so that people could find out.
Yeah, I wish you had reviewed my paper. So we submitted a paper years ago that said poliotropism is not controlled by the internal ribosome entry site, the iris. Okay? Mm-hmm. So we sent it to Nature, and they said, this is a negative result. We don't want to publish this. Eventually got published, but actually I also went to the PLOS journals, and they said, we don't publish negative results. So this kind of broad... hey, Alan, weren't we talking about absolute statements in the pre-show? I mean, that's ridiculous. We don't do this or we don't do that. You have to look at every case individually. But, I mean, eventually it was published, and I thought, as you said, Kathy, it prevents people from repeating the same experiments, if anything, right? Mm-hmm. So I find this a little funny because I would like to see a paper also saying we have the receptor, but as you said, there's some value to this too. So
well, and in the Nature News story on this, they comment on this ACE2 receptor paper, and then apparently that team declined to elaborate on which receptor it does use pending a separate publication.
So they know already, huh?
So maybe they know already, or they have preliminary data already. I don't know.
Mm. Of course, there are many ways to find receptors, and we've talked about a lot of them here on TWiV. So it will be interesting to see when they do it. We'll do that paper, right? That'll be interesting.
Sure.
See what it is.
So both of these coronavirus EMC papers are in MBio, and that's another open access journal. So everybody could go look at those.
Yeah. Yeah, that's really nice. ASM decided to make an open access journal, which is really cool. All right. Did everybody like that?
Mm-hmm.
I thought we should update this Corona. So there you see what's
Yeah,
happening.
definitely.
I think we should also do a TWiV on influenza very soon because that's in the news now.
Oh, yes.
And we should make everything clear what's going on.
Yeah.
All right, let's do some email. Kathy, can you take Johan's?
There we are. Okay. Johan writes, "Hi, love all your shows. I have a question regarding Janine Roberts, if you have heard of her. You can take a read at," and he provides a link to a paper that I think was from 2004. He says, "I know you're the king of polio, so I'd love to hear your version of it. Thanks, Johan from Sweden."
The king of... Who was just the king of something here? Somebody else was a king, right? We just read an email. Anyway. Harmit is the king of genetic conflict.
That's right.
I'm not the king of polio, but neither is TWiV. But we can certainly comment on it. What do you think about it? You read this, Kathy, right?
Well, I didn't read it. I started reading the first couple of paragraphs, and then I just started skimming it. And my take on it is it's written in a semi-convincing way, kind of in the format of a Discover magazine article or something, but it's just full of mistakes, and it doesn't cite the rigorous science experiments that have been done. So I think it's dangerous for a layperson to read something like this if they're not skeptical or they don't have a way to get at the real truth.
Yeah. The title is Polio, the Virus and the Vaccine. Her thesis is there is a massive historic evidence that suggests it is not caused by a virus but by industrial and agricultural pollution. Okay, so you take a little isolated bits of information on their own, as you said, without the real science, and this sounds interesting to a layperson. But in fact, we know from so many lines of evidence that polio is caused by a virus and that we can vaccinate and prevent it. There's just no other scientific way to look at it.
Pretty much fulfills Koch's postulates that it's an infectious agent.
Yeah. So
we know the cause of polio. This is not up for grabs, but this is an example of an entire genre of stuff that's out there, for want of a more family appropriate term, on the web, and if somebody decides on a theory for whatever reason, either because it fits their political leanings or they just like to believe these sorts of things, and then goes shopping for evidence for it and cherry picks little bits and pieces from here and there and puts something together and says, this is the real story. And of course, it's not the real story, because if you actually looked objectively at the evidence and started with the data and then went to your conclusions, you would reach completely the opposite.
Yeah, for example, that she talks about the isolation of poliovirus by Landsteiner, in 1906. And she said, they took a sick chimp and ground up its spinal cord and infected another one. There were so many things in that spinal cord that could have caused the disease. Well, it was passed through a 0.2 micron filter, which you neglected to mention. Which only passed it. And here she says, at the end of 1952, the spread of polio was not affected by the closure of schools as it should have been if the disease was infectious. So that statement on its own would cause someone to have pause, right? But in fact, we know that asymptomatic people spread the disease, so it doesn't help to close schools.
Right.
So there's just over and over again, there's all this wrong stuff. So, yeah, as you said, Alan, this is a very common thing to do in all sorts of science areas, right? To list all this stuff and ignore the real science. So we don't think very much of it at all, because polio is clearly caused by poliovirus. Eradication is almost there because of immunization.
We still have plenty of agricultural and environmental pollution. I wouldn't deny that that's a problem, but polio is going away. So
remember, there's association and causation, and that's what you have to think about. And
this doesn't even fit. It doesn't even fit as a correlation.
Right.
Because as I said, we still have pollution, and we don't have polio in most places. So it just doesn't work at all.
No. I have a book here on my shelf that someone sent me years ago, and says the association of poliomyelitis with high sugar glucose. Same sort of thing. So
Yeah.
if you had high blood sugar, you got polio.
That was a whole book?
Well, it wasn't very big. It was about a half-inch thick.
Wow.
I must admit I did not read it. But someone sent it to me years ago, mail with a handwritten note. Please read this. I think it's important for you to understand that this is the cause of polio. Okay.
Well, and in fact, historically, even in the scientific literature, before the cause was known, there was a lot of speculation about what it might be. And
sure.
These theories did have currency at one time. It's just they don't hold.
They don't hold any longer, and you have to let it go. And it's a very long article, and this lady spent a long time doing this, but you shouldn't spend a lot of time reading it. It's really not, unless you want to be an expert on debunking these sorts of things.
But that's not even worth anyone's time.
So, Johan, we don't think much of it, but thank you for sending it to us. We do appreciate the opportunity to comment on these things. Okay, the next one is from Peter, who writes, Dear TWiV team, there are many cases of norovirus in the UK this winter, estimated at 100,000 cases over the Christmas period, with the number of confirmed cases 72% higher than this time last year. Gives a link to an article about this. I have read that following a norovirus infection, a person's immunity only lasts for around 14 weeks. I wondered if this is solely down to the rapid mutation rate of the virus, or does a person's immunity to this type of virus decline rapidly over a short time? So I sent this email to Christiane Wolbus over at your university, Kathy, and she wrote, both viral evolution and inability to raise long-term immunity are to blame. There are old papers from the 70s showing that human volunteers infected with exactly the same strain of virus will be reinfected after 1 year, but not if the interval is 14 weeks or less. So short-term immunity develops, but not long-term immunity. Also, noroviruses show antigenic drift akin to what is seen with influenza. Wow. So those are volunteer experiments where people drink fecal extracts because you can't grow the virus in culture. How much would it cost for you to do that, Alan?
I don't know if I could put a price on that.
So these are medical students who are...
Yeah.
Wasn't this the thing that Michelle Osmond said that she didn't get sick?
Yeah.
Wasn't it this virus? Yeah.
Yes.
It could be. Yeah, some people don't get sick.
I think she participated in one of these studies.
Yeah.
Also, check out this entertaining read about noroviruses, which for a general news article is quite accurate. This may be a fun read for your listeners, how not to catch the norovirus.
Yes.
It's from The Guardian. Hope all is well. Best wishes for successful 2013. So there you go. Right from the expert.
Yeah, the article is pretty hilarious. And in fact, Christiana gave a departmental seminar and used part of it as her introduction and had the audience just in stitches.
How not to catch norovirus.
The lead is great. The laugh-a-minute pro-celebrity puking bug known by the street name Norovirus continues to squirm its way through the population.
Want to avoid Spumageddon. Here's a simple guide. Spumageddon is a nice episode title, too. Wow.
It's like Piers Morgan, but invisible. Oh.
Alan, would you take the next one from Dallas, please?
Okay, Dallas writes, Vincent, the question of life seems to keep coming up. The existence of metabolism has been stated as a significant difference. However, metabolism can be viewed as a dynamic chemical kinetic problem in a system that isn't in thermodynamic equilibrium. Other pure physical chemical systems also appear lifelike, such as various reaction diffusion systems, mineral growth systems, etc. And he sends a YouTube link. I have not seen this video yet, and I'm not going to click on it now because we'll end up with the audio. But what is it?
It's actually quiet. I think there's no audio. And it's these solitons that are self-reinforcing solitary waves. But these look like little cells kind of moving around in Brownian motion at first, but by the end of it, they're all going off in the same direction, or whatever that means.
Okay, so it's a chemical reaction. It's a chemical diffusion system that gets these things moving around. Right. Kind of mesmerizing, actually.
Mm-hmm.
So this statement metabolism can be viewed as a dynamic chemical kinetic problem in a system that isn't in thermodynamic equilibrium. Is that correct? I suppose it is, right? One way of looking at metabolism.
I guess. Right.
Yeah.
So then if you define it that way, then you can include a lot of things doing metabolism that aren't really living. But that's just one of the properties of things that are living, right? So these solitons don't have other properties. Evolution, right? So,
Yeah, they don't reproduce.
so.
Right.
Speaking of living, when I got this email, I went to see the Are Viruses Alive poll. And I think, Kathy, you had said a couple of weeks ago that it was down.
You discovered that, yeah.
So I went and, I was so sad because we had thousands of responses. And then I had it at surveymonkey.com, okay? And for some reason, they took it down. And now you're limited to 100 responses, otherwise you have to pay $20 a month. And for one poll, I didn't want to pay $20 a month, even though I think it's an important one. So I did find another site that is really quite good, and it's free for unlimited answers. So it's back up at virology.ws/r-viruses-alive.
Okay.
And I think it's actually a nicer looking poll, and you can answer it right there. And then there's a link at the bottom of the page where you can see the results.
Oh, I didn't see that. Okay.
So it's new, and everyone, if you feel like it, go back, and we'll build up to a few thousand answers again. But it'll take time. I'm going to give it to my course in a couple of weeks, too.
Oh, so
Yeah,
this is a brand new version, so I should go ahead and answer it.
Yeah, answer it. This is a brand new.
Okay.
We have 10 responses.
Everybody can reply, even if you did the previous
Yeah, it's brand
poll.
new. I got, are viruses living? Yes, no, maybe, I don't know. So far we have 2 yes and 8 no. So do it. Everyone go over and do it. It'll take you, 2 seconds, 10 seconds.
Wait, didn't you have any answers to the... somehow I thought it was different. So if I say an answer, if I vote again, okay.
Yeah, you can do one answer, and then you click next, and it gets registered when you click next, and it says thank you.
Okay.
It's a little different from the other one, but that's all I could do for free. All right. Kathy, can you read John's?
John writes, Dear Twivers, I can't thank you enough for the wonderful podcasts. When I graduated from college, I used to think that people who didn't read the New York Times, the Wall Street Journal, or the like were hopelessly ignorant of the world around them. Given the explosive growth in high-quality podcasts such as yours, I now consider people who get their information primarily from the popular press to be the ignorant ones. I also love Vince's lectures on iTunes U. Keep up the good work. John.
All right.
That's nice.
The last one for today, second to last, is from Will. I have a question regarding rabies virus. First of all, I heard that once the virus enters peripheral nerves, it is sequestered from the immune system. Is this true? And if so, how? I heard that rabies virus is intracellular and neurotropic, meaning it hides within neurons. Is it that antibodies created by the vaccine are not able to enter into neurons in order to kill the virus within? I've heard elsewhere that antibodies can enter cells in order to defeat the viruses within them, but I've read so much contradictory stuff elsewhere that I don't know what is true. This seems like a pretty reputable blog. I was hoping you guys could set the record straight. So Will sent this to Virology Blog. Then he sent it to TWiV, and then he posted it on Facebook.com/thisweekinvirology. I said, I'm going to answer you on TWiV this Friday. Take it easy.
He really wants the answer.
Wants the answer. I guess he has a test or something. So then he said, I know antibodies can enter neurons where rabies viruses are hiding, but how do the antibodies know the viruses are in there? Is it just by chance that the antibodies enter the neurons and find the viruses? And also, I know it slows the spread of the virus in the CNS, but what about peripheral nerves? So, you can get bitten by a rabid animal, get rabies in you, but you have time to be immunized, which means you can get treated post-exposure, and it works to prevent rabies.
Yes, so Will, if that's the source of your urgency, go to your doctor and get the shot.
Yeah, if you do have rabies. But if it's for a test, we're going to talk about it a little bit now. So my suspicion is that the antibodies probably either bind to nerves or peripheral nerves or nerves otherwise, or get in via endocytosis and neutralize the viruses within. there is some evidence using rabies that you can clearly get clearance of the virus from the CNS by adding antibodies to animals. And how they do that is not really understood. Now, in another virus, alpha viruses, a number of years ago, Beth Levine and Diane Griffin showed that alpha viruses, which are neurotropic, can be cleared by adding antibodies to the infected animal. And the antibodies don't actually get into the CNS, but they bind to nerves and somehow restrict virus replication in that fashion. So we don't have as much data for rabies as far as I can tell, but I think either the antibodies get in or they bind to the nerve and restrict replication.
So I had a discussion with Beth Moore, who's teaching virology with me, and she's an immunologist. And she said, yeah, there are examples of antibodies that work once they're internalized, but this isn't really believed to be the major mechanism, and it requires a cell type that can be phagocytic. And that 1992 paper with rabies used neuroblastoma cells. And antibodies, of course, are most effective against extracellular antigens. And so her thought was maybe that where the antibodies are effective is between nerves, when the virus is in the junctions between the nerves extracellularly, perhaps. Right. That was her thought.
So if you are, let's say you're bitten on the hand and the virus has entered your nerve ending there and it's traveling towards the CNS. So it has to go through multiple cell-cell junctions, right? It's not a single cell all the way to the brain, right?
Right.
Yeah, so that would make sense because the virus does have to cross, and so that's where the antibodies could work, yeah. I mean, clearly for Synbis, this alpha virus, the antibodies bind to the nerves, and that knocks down virus replication. But I don't know if that happens for rabies. There's no evidence for it. But basically, we don't know the answer, right?
Right. Yeah. Well, and also, what she mentioned to me, and it rang true, is that the vaccine is effective if it's given fast enough, and also depending where the bite by the rabid animal was. So if it was your foot, you have a better chance than if it was your shoulder.
Right. Or your face,
because there's less distance to, and so that, again, might argue that the antibody treatment has to hit, during that transmission phase, has to act during the transmission phase when the virus might be between cells.
Yeah.
Anyway, her bottom line was that the body's immune system just doesn't rev up fast enough to prevent the rabies spread. And why that is, I think, is still a big question.
Well, it could be hidden, right, in the nerve. Once it gets in the nerve, then you're not making an immune response to it.
Right.
Because you can be immunized peripherally with rabies vaccine, after a bite, and you will make antibodies, and those will prevent entry into the CNS. So I suspect that Will is right here, that the virus is hidden in the CNS or in the peripheral nerve once it gets there.
Right. And I guess what she was referring to is that the virus replicating in the muscle cells isn't somehow generating an immune response.
Yeah. That's
Why
a good
is
question.
that?
Yeah. I wonder if it's taken up really quickly and, it doesn't replicate extensively in the muscle. Yeah, that's a good question.
Yeah, it should be able to get to a nerve pretty quickly, but you'd think that the initial insult would stimulate something.
Yeah. Apparently not. But did she mean to say that peripheral nerves don't do endocytosis?
I don't want to misquote her on that one way or the other. I don't think she was.
Yeah, because I know for polio, one of the models is that at the neuromuscular junction, the virus is taken into the nerve by endocytic pathways, and it travels up in a vesicle by motor transport. So I think there is some endocytic activity, and so antibodies could get in also that way.
Right.
So I don't think we could rule that out.
Right.
I think it's an interesting question, Will, and if you have a test, there's no right answer, you can do this speculating.
Mm-hmm. Right? Yeah, and the reason you're finding contradictory information is that the information we have is contradictory.
Yeah. All right, our last one is actually a very interesting tweet from Hurricane Alicia on Twitter. And she wrote, Voodoo Virology, how can Roche withhold its data. And she linked to a Forbes article by a Yale professor named Harlan Krumholtz. He's an epidemiologist at Yale. And it's called The Myth of Tamiflu, 5 Things You Should Know. And it actually covers something we talked about a long time ago, that the data for the clinical trials on Tamiflu, which were done by Roche, are not all available. So he points out this Cochrane review, which is an organization that does meta-analyses, has requested additional data to try and find out if Tamiflu, the antiviral against influenza, has certain effects beyond what it's licensed for. I mean, we know that if you take it, it will shorten the time of symptoms, but it doesn't do a lot of other things like prevent hospitalization or prevent transmission. And so Cochrane said, give us the data, and Roche said, no, you can't have it. And so Alicia said, how can that be? Well, so I asked someone who works in the pharmaceutical industry who will not be named, but that individual said if they did this trial together with, for example, a medical center, it is not their data to release. They can't just say, here's the data. And individual patient data, which they requested, is usually never provided. So what they give to the FDA is a subset of that. And then finally, this individual said all Cochrane wants to do is publish something using data that they didn't pay for and which cost a lot of money. And so what do they think they are anyway? So there is some precedent. I'd like to hear what you think, because I'm sure you've thought of this, Alan, before as well.
Yeah, it's a tricky situation. So it's very easy to take the stance that all data for all clinical trials should be disclosed completely. But as soon as you start looking at what that would entail, first of all, you don't want any personally identifiable information on the participants to be disclosed. That can't become public. And so the general public will think, oh, okay, so you just strip the names off the medical records. Well, it's not that simple.
Right.
If you know that somebody is a 60-year-old virologist living in New Jersey who works in New York City, you can really narrow down the population of people you're talking about here. So you've got to remove additional information from the records, and then at a certain point you remove so much information that it becomes scientifically useless. So you can't just publish raw data from a clinical trial. The thing that you could publish would be the stuff that you would put in a paper, right? The actual publishable data that you did this trial to collect. And I think that's what the Cochrane group is asking for here.
They want all the data.
Yeah, well,
everything that wasn't published. Right.
Right. But presumably not. They don't want to know how many milligrams of Tamiflu Joe Smith in such and such trial got. They want to know, statistically, how many patients did you enroll? What were their background health conditions? Looking at the population level data in the study, as you would do if you were publishing it in the New England Journal, for example. And there could be reasons why Roche can't or is unwilling to release those data that have nothing to do with trying to cover anything up. On the other hand, we can't say definitively that they have legitimate reasons to withhold the data unless somebody has seen the data outside of Roche.
So Cochrane asked Roche for full clinical study reports with study protocols, reporting analysis plan, statistical analysis plan, and individual patient data so that they could more fully determine what could be concluded. All right. Now, it's very easy to see Roche as the bad guy here, the way this is worded. But in fact, they've already submitted the data to the FDA. It's been approved for the alleviation of symptoms. If they really thought that it was good for something more, they would have applied for it.
Well, the issue, I think the concern is that maybe these other studies showed that it wasn't as good for alleviating symptoms. Right?
Yeah, okay.
So if Roche collected additional data that didn't support what they wanted to prove, there would be a strong motivation to not release those data.
Yeah. So are you saying they selectively release data that would get the drug
I'm not
approved?
saying that at all. I'm saying we can't tell from the information available.
Yeah.
Of course, a company is going to choose their best and most definitive trials to submit to the FDA.
Right.
The question is, are the other data bad for unrelated reasons? Did something happen to the trial? Maybe there was a bad flu epidemic that somehow messed with what was going on, or there was maybe it was a really mild year for flu when they did the study. These flu studies all the time. So maybe they're not releasing the data just because the data are crummy.
Yeah. No. I mean, look, they've chosen not to. Maybe why should Cochrane be the ones to analyze it? I mean, maybe they'd screw up and not get the right answer. I don't sort of blame Roche for not wanting to just release it out there. After all, this is a privately funded trial. It was not a government-supported trial.
Yeah.
So if you guys want to have a for-profit pharmaceutical industry, this is the price you're going to have to pay. They don't have to share it. And you know what? Go get vaccinated. Don't take Tamiflu anyway. You should just get the vaccine. Only 40% of the U.S. gets immunized against flu every year. That's crazy.
Yeah.
Yeah.
You got to get it. You guys get your vaccines, by the way?
Yep.
You, Alan?
And Alan?
Sound gone?
Yes, I'm here. Can you hear me?
Yeah.
Yes.
Yeah, I did get it.
Okay.
Yes. Yeah, I told my class, because actually I showed them or told them that the map for the United States and the flu prevalence, this week Michigan is red, last week it was yellow, the week before it was green, red being the worst. And we got mail from the health system saying, vaccine's still out there, go get it. So I don't know how many students will do it, but yeah.
Yeah, a lot of people don't, but what are you going to do?
Yeah.
All right. Let us do some science picks of the week. Alan, what do you have for us today?
I have a film that was made by somebody you may know, somebody I know. This is Stephen Curry.
Oh, yeah, I do know Stephen.
Yeah.
Crystallographer.
A crystallographer, exactly.
He trained with Jim Hogle.
Exactly. So I met him at a Gordon conference years and years ago, and I recently found he was on Twitter and was following him, and he plugged this film that he made, and he made it about a year ago, in September 2011. He got a small grant. The videography and the editing is quite good. And it's a short, about a 25-minute film. What is it like to be a scientist? It's aimed at school teachers, so they can show this to their classes. And he just takes a really cool approach. He interviews several scientists, and he asks them a bunch of questions about how they got into science and what is their work like and what's appealing about it, and does every scientist have a Nobel Prize, and just really, really kind of humanizing what it's like to do science and what kinds of people go into it. So I thought it was a really, really cool outreach.
It's over a year ago it was released. Yeah. Wow, I missed this because I do follow Stephen, but I missed it. That's cool. Nice. Check it out, folks. Mm-hmm. Kathy, what do you have?
I picked Humorous Units of Measurement. It's a Wikipedia entry, and a friend of mine sent a link to just one of these and said, "You wonder why I found this?" And then about a day later, he wrote and explained why. But it's got the smoots, which I had heard about on Car Talk. Various interesting, weird measurements. The Beard second, the Sagan, the Bottlesworth, bogosity is the measure of bogosity is the Linat. Is that how you say it? Linat. The Fame non-conventional. This is in the non-conventional measurements. The measurement for Fame is the Warhol. So it's just a funny little thing in Wikipedia to check out.
We have some in science that now totally escape me, like minimal publishable unit.
Yeah, at least LPU.
Least publishable unit. Yeah, you go to a conference and people accuse each other of, hey, you have a half an LPU in that paper. That's cool. Gosh, some of these I've never heard of.
I like the barn as a unit of area.
In fact, the barn, I think the barn is the one that my friend first connected with. No.
Yes, and it is an actual accepted standard international unit that was originally referred to as, like hitting the broadside of a barn. It's actually a very small thing, but then there are smaller subunits of it that have been called the outhouse, which is 10 to the minus 6 barns, and the shed, which is 10 to the minus 24th barns.
Cool. Nice. My pick is a article. It's a commentary in Nature by Cameron Nalon, who I met a couple of years ago over there in the UK. It's called Science Publishing Open Access Must Enable Open Use. So the idea here is that, we need open access, but beyond that, you need to be able to take the data and do something with it. As he said, a lot of open access journals don't allow you to do that. He has a really good example here. If, for instance, you wish to index all the gene names in a set of papers, put them on a website, translate them, use text or images in a summary, or even just print out several copies of the papers, you are limited to a small set of around 500,000 articles that carry a Creative Commons license. So, for example, that's why. Because you can't do that with all the articles that you would want. So he's just saying we need to be able to manipulate and redistribute the data, which I think is good, especially for genomic data and that sort of thing. So I think it's an interesting article. And of course, we're not even at the open access point uniquely so far, right? I mean, we're getting there, but this is he thinks is the next step, which is kind of an interesting read.
Yeah.
So, because I've never really understood it, I tried to figure out what the Creative Commons means, and maybe one of you guys can explain it in more lay terms to me. Something that has a Creative Commons license versus something that's copyrighted or whatever.
You can go ahead, Alan, do that.
Yes. Creative Commons is a specific type of license that the courts have now ruled is legally binding, that explicitly gives rights to the user, to the reader, to the consumer of the content. So I'll give you an example. Let's say I take a photo of something and I post it on the web. If I put absolutely no additional information on that photo, then by law, it is copyrighted from the moment I created it. And I am entitled to copyright to that image. If you want to use that image on your website, you can't just grab it and put it there. You would have to ask me for permission in order to legally reuse it. If I put a Creative Commons license on it that specifies the conditions under which you can reuse it, if I say you can reuse this in any form as long as you give me credit, then you don't have to ask me for permission anymore. You can take that picture, put it on your website. You can if I, depending on the version of the Creative Commons license I've chosen, if I say it's okay to use derivatives of it, you could take a crop of that image or cut out some piece of it and use it in a collage or some other work that you're working on, or you could use it as a backdrop on a news show or whatever you want to do with it. And all of that would be permitted under this blanket Creative Commons license that I have put on it.
I see.
So you do have to specify that you want the Creative Commons license, because otherwise, under copyright law, if you just set it out there, even if I made a statement on my website to the effect of, people can use whatever they want, there would still be copyright complexities in somebody borrowing stuff from my site. So the Creative Commons license is an actual legal contract that puts it out there and specifies the ways in which it can be reused.
Yeah, the PLOS stuff is all Creative Commons.
PLOS stuff is all Creative Commons. Most open access journals insist on something like that. And there are other little fine points of this. Anything created by a government employee as part of their employment is automatically in the public domain. So in that case, you don't have to ask for permission. And in fact, the Creative Commons license doesn't even apply. It can be used by anybody for anything.
Yeah. Yeah, they go to creativecommons.org. You can select your license.
You can pick exactly what type of license.
Now those all make sense to me, but I didn't get them with respect to copyright. And so that's very helpful.
So then you put a little badge at the bottom of your website. So all my websites, blog, podcasts, my lectures, they're all Creative Commons attribution only, that you can do whatever you want with it. Just say that you got it from me.
I see.
Sometimes you don't have to attribute. Sometimes you have to do other things. It depends what you want to pick, but I want.
You can say whether you allow reuse for commercial purposes or not, and all that sort of thing.
They can make millions of dollars off my stuff if they want, as long as they say they got it from me.
Right.
All right. We have a listener pick from Tarwin. I found this great game which seemed to go viral a month ago called Plague Inc. It's an iOS Android game where you play the part of a plague, viral, bacterial, or parasitic. They seem to have the whole Twix table covered for now. The game centers around upgrading your pathogen by mutation to kill more people. The aim is to wipe out the human race, so you can't get too strong too quickly or you'll simply wipe yourself out too. Very interesting look at pathogens. And even if it isn't 100% accurate, it does give some great insight for casual players, almost educational. So it gives a link to it. Thanks again for the amazing work you all do. Every listen, I get closer to dropping what I do and starting a whole new career, currently a web developer. Taran's a web developer in San Francisco. He's at the company Touch My Pixel, which makes apps. It's cool.
Cool.
Reminds me of Pandemic, which my son reviewed way back on TWiV number 7. Same idea. You want to wipe out the human race, but that was a computer game back in the day before there were these apps on mobile devices. Same idea. Pretty cool. I might pick it up, but I have too much else to do right now. But check it out. Thanks, Tarwin. It'll do it for TWiV 215, which you will find at iTunes at twiv.tv. And if you like us, please go over to iTunes and leave a comment or a rating to help keep us prominent there. And don't forget our Facebook page, facebook.com/thisweekinvirology. We love to get your questions and comments. Send them to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks for having me. It's always fun to learn things here.
Glad to hear it. Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.